Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PU first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, we shall start today's session. In our today's session, we are going to discuss about linkage and recombination. Okay, about this linkage and recombination which we have started in our previous session, that is the experimental verification of chromosomal theory of inheritance, which was given by Thomas Hunt Morgan and his colleagues. Okay, so after that we are going to discuss in that same the experimental proof or the way he has verified, that we are going to discuss in this heading called as linkage and recombination. Okay, here we can see that Morgan is what? Thomas Hunt Morgan. Thomas Hunt Morgan carried out several dihybrid crosses. Dihybrid crosses, hope you remember, which we have discussed about by Mendel. Okay, which was given by Mendel, dihybrid crosses. That dihybrid crosses, here he has crossed with Drosophila. As we know why Morgan chose Drosophila, right? So Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. Okay, to study the genes that were sex-linked, the genes which were sex-linked here, which means they were knowing that sex-linked means it is the chromosome which is present on the X chromosome. Okay, they have studied about the genes which were present on the X chromosome. Okay, the crosses were similar to the dihybrid crosses, which means that what we have discussed about dihybrid cross, those were similar as it was carried out by Mendel in pea plant. For example, Morgan hybridized yellow-bodied white-eyed females, which means here Morgan has chosen female Drosophila, which was yellow-colored body and eyes were white color. Okay, this was crossed with brown-bodied with red-eyed males. Okay, males were red-eyed and brown color body. These have been intercrossed and they found their F1 progeny. Okay, their F1 progeny was self-crossed as we did in a dihybrid cross which was given by Mendel. He observed that the two genes did not segregate independent of each other. He observed that these two genes, they were not segregating independent of each other. According to the Mendel, the two genes were segregating independent of each other, which we have discussed in law of independent assortment. So this was total deviating, and the F2 ratio deviated from 9:3:3:1. Normally, as we find 9:3:3:1 is the ratio, phenotypic ratio of dihybrid cross, but here you could not find that. Okay, it was totally different. Okay, why it was different and all, we have here theoretical part. First I shall discuss about theoretical part, then we shall discuss about its experimental proof. Morgan and his group knew that genes were located on the X chromosome. As we have said, when it is sex-linked, it is on the X chromosome. And he saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, when the two genes are present in a dihybrid cross, they were situated on the same chromosome, the proportion of parental gene combinations were higher than the non-parental type. Okay, the proportion of parental gene combinations were much higher than the non-parental type. How it is possible, we shall see here. Okay, if I am showing this as one of the chromosome, okay, according to your sentence, when two genes in a dihybrid cross were situated on the same chromosome, two genes, if I am showing, this is one gene, this is another gene, these are the two genes. Okay, were situated on the same chromosome, the proportion of parental gene combination, now these are the parental, the proportion of parental gene combination were much higher than the non-parental type. This combination will be more than the other. If we are talking about one more chromosome and its genes are present here, now their combination, this parental gene combinations will be more rather than getting exchanged. Okay, and this exchange is nothing but recombination, which we have discussed about as crossing over in first year. Okay, you have about crossing over in meiosis. So this crossing over or this recombination will be less. This is non-parental type, these are parental type. Okay, the proportion of parental gene combinations were much higher than the non-parental type. Parental gene combinations are higher, non-parental type are less. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage. Okay, Morgan, why he said this? Because of physical association. When these two genes are present nearer to each other, there is a association, physical association among these two. They are getting attached with one another. They are linked with one another. Okay, if these genes are present far away from each other, then their physical association also will decide whether it has to undergo recombination or crossing over or not. Right? So physical association or linkage of the two genes, and coined the term linkage, which means who coined the term linkage? T.H. Morgan, Thomas Hunt Morgan. He discovered, or he coined the term linkage to describe the physical association of genes on a chromosome, and the term recombination to describe generation of non-parental gene combinations. Linkage is for what? To physical association. Linkage is for this. This is physical association of genes. Okay. Recombination is what? When they are getting exchanged, crossing over. Okay. Non-parental gene combinations. This is about recombination. So linkage and recombination, these terms have been given by whom? That is Thomas Hunt Morgan. Okay. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were tightly linked and some were loosely linked. Okay, so tightly linked means, as I am showing here, this is the chromosome. If these are present nearer to each other, genes are present nearer to each other, they are tightly linked because distance between them is very less. So linkage between them will be stronger. Okay, when linkage is stronger, then recombination will be weaker. Recombination will be weak or weaker. Linkage is stronger because there is a strong bond. When you are keeping two magnets, okay, two magnets with the same pole, with the opposite poles here, something like this, and what will be the attraction, force of attraction, what it will be there, and what will be the force of attraction in these two magnets which are kept far. Okay, so here what you can find, there is a strong linkage if genes are present nearer to each other. If there is a linkage is stronger, then recombination, chances of recombination will be less. Okay, if the genes are present far away from each other, in that case, if I am showing like this, one gene is here, another gene is here, the distance between them is more. So what you can say in this, linkage is weaker, then what next? Recombination is stronger. Okay, or where there is a more recombination, less linkage. Where there is a more linkage, there is a less recombination. Okay, this is how you can remember this. To show this tightly linked and loosely linked, they have given about two of the experiments which I shall show you. And before that, proceeding further, about the distance between the genes was studied by Thomas Hunt Morgan's student. His student was Alfred Sturtevant. Used the frequency of recombination between gene pairs on the same chromosome as a measure of distance between the genes and mapped their position on the chromosome, which means what we are discussing here, that genes are present here and here. The distance between the genes was studied by, okay, Alfred Sturtevant. Alfred Sturtevant is who? He is the student of Thomas Hunt Morgan, and he gave about the linkage map or gene mapping. Okay, gene mapping was given by Alfred Sturtevant. He was the student of Thomas Hunt Morgan. He studied the distance between the genes, how it matters, whether it matters, how it matters, if it is present on the same chromosome or on a different chromosome. Every detail has been studied by him. Okay, this is about that linkage map and Alfred Sturtevant. You have about that one in your textbook here on the page number 83. Okay, now here in the next page you can find in your textbook that this kind of crosses have been given. Okay, these crosses they have given as cross A and cross B for linkage. Okay, these are the dihybrid crosses. Now this will be easier if you know about Dihybrid Cross, what was given by Mendel. Okay, so I shall try to show you all here. First, yellow-bodied, white-eyed. What was this? Yellow-bodied, white-eyed. It was female, right? We have just seen that, yellow-bodied, white-eyed, and it was crossed with whom? It was crossed with wild type, that was brown bodied and red-eyed males, I think. Yes, where it is? Yellow body white-eyed females, brown body red-eyed males. Okay, brown body red-eyed males. Okay, these are females, these are males. For this, it has been mentioned as wild type in your textbook. See here, this is the male, this is female, this is yellow and white, and this is wild type. Okay, now these are what? Parents. The chromosome, X chromosome, because females are having 2 X chromosome, XX, so same chromosome has been shown here. And for genes I am showing it like this, which is having YW, and here YW. Okay, similarly for brown body red-eyed males, because it is a male, one will be X chromosome and another one will be Y chromosome, which means both the chromosomes differ from each other. So in this you will find Y plus W plus. Okay, so for this genes it has been given it as Y plus W plus. For this it has been given it as YW. Okay, now what happens in this? In this, if you imagine they produce, now what we'll write? We'll write about the gametes. Okay, gametes will be the same, right? So gametes, this will be acting as one gamete. I shall use pencil here. This will be acting as one gamete and this will be acting as another gamete. I will show you those gametes also. If not, you will get confused. Okay, production of gametes, which is not there in your textbook. You will find something like this. Okay, YW, and just roughly I'm showing YW. Same one you are going to draw it here. For this we shall show you like this, Y plus W plus, and another one is something like this. This is Y chromosome, right? This is how we are going to show you. These are the gametes. Now F1 generation. Okay, what you have in F1 generation, we are taking this, we'll take this and cross with this, right? We'll get how? First we shall draw this, we'll get YW, and we shall draw this. What genes you have here? Y plus W plus, right? Okay, then you have this one and we are crossing with this. I am not taking this because both are similar, both are same, so I am taking only one and I will show you. Okay, you will find YW, and here this one, it is something like this. Okay, now what is this one? This will become wild type, Y plus W plus. This is wild type. Wild type means what? It is brown bodied and red-eyed, we call it as wild type. Okay, now this we call it as yellow white, yellow white, yellow bodied, white-eyed, which is the characteristics of this. Now why we are giving this one as wild type? Which means this Y plus W plus is dominant. Okay, Y plus W plus is dominant, to show that we are giving it as superscript as plus sign. Okay, now this is how you are going to get. Similarly, you will find that one in your textbook. Here in your textbook they have given YW, YW, Y plus W, Y plus W plus, and wild type. This is how it has been given, and what they did is directly they have jumped to the F1 generation, where they have not shown the gametes of it. So when we are finding the F1 generation, what you find? We took one. Okay, because both are similar, we are taking only one. YW is crossed with Y plus W plus. We got this, which means yellow with the blue. Okay, now yellow with the purple. Okay, you find this. So one is wild type, another one is yellow white. Hope you got this. Now after that, here next stage, what you will do next stage? As per we know what we are doing, we are writing its gametes and crossing with that. Am I right? So what we'll write? Gametes of F1 generation. Gametes of F1 generation. So in the gametes of F1 generation, what you will get? Will show like this, YW, is it? And another one is something like this, Y plus W plus, right? here we have this, its gametes. Its gamete are, one is YW, another one is, next one is yellow, this one male. Okay, a Y chromosome. Now first we shall give for the parental type and then recombinant type. Okay, parental type means as it is present in the parents. Fine, I shall show you the other recombinant type also. First we shall see the parental type. Now parental types, what we'll do here, now we'll cross this. We shall take this one, right, and cross with it. Yes, we are going to do this. What we'll get, YW, and from here YW, is it? Okay, then we are taking this and we are crossing with this. Simultaneously if you draw, it's better to understand. Okay, this chromosome is here, right? We got this one. Now after this we shall take this and cross with this, Y plus W plus. Okay, and another one we have YW, right? Then we have this one is Y plus W plus, and another one we have here is the male chromosome. Okay, so this is actually how we do normally. Either you can use a checkerboard or directly you can do this. So what we have, YW means now this is yellow white, is it? YW and a male, right? YW and a male is yellow white. Y plus W plus and YW, because it is present Y plus W plus, then it is a wild type because it is dominant. Okay, so because it is dominant, it is a wild type. Then comes this with the Y plus W plus, then definitely this is also wild type. So we got two wild type and two yellow and a white. These are the parental types. You can find here in your textbook parental type and recombinant type. This line is a parental type and this side is a recombinant type. In a parental type, what they did, okay, first they have taken about this blue one, that is Y plus W plus, and that Y plus W plus, it has been crossed with this male. Okay, first they have taken this with this male chromosome. You can find it here. Then what they did, they took YW and crossed with male. Okay, first with blue with a male, then yellow with a male, blue with male, yellow with a male. Then you can find what they did, they took blue and crossed with yellow, blue with yellow, and yellow with yellow, yellow with yellow. Okay, it is same as what we have got here. These are the parental type. Now recombinant type means what? In recombination, there is an exchange of chromosomes, exchange of crossing over take place. Now we were here, right? We were here. We have given this one as F1 generation, wild type and yellow white. These both you can find here in your textbook. One is yellow color, another one is blue color. If they are undergoing crossing over, if I am showing these chromosomes here as I have shown earlier, now YW, I am taking this YW, which YW? This YW, yellow color, and another one more chromosome with Y plus W plus. These two chromosomes are there. Okay, now in this crossing over does not take place because dissimilar chromosomes are there, right? One is X, another one is Y, so no crossing over takes place. Only crossing over takes place here in these two chromosomes because both are X chromosome, both are similar chromosome. Now when crossing over takes place, what happens? Right, when crossing over takes place, it happens something like this. exchange of chromatids takes place. For one we are giving it as YW, for another one we are giving it as Y plus W plus. Now what happened in this? These two are getting exchanged, something like this. Okay, when they exchange, then what happens? Here the chromosomes, when they get separate, these two chromosomes, right, these two chromosomes which were present, one will be having W and Y plus, another one will be having Y and W plus. Hope you understood this. After crossing over, imagine that this chromosome is blue color, okay, W is yellow, okay, this one is yellow color and this one is blue color. Now small part of it, this has become blue color, yellow with blue and blue with yellow, like that, as it has been shown here. Okay, now these two genes here, these two genes, now WW is here. What happened is Y plus has gone to that side and Y has come to this side, which means Y plus W and YW plus. In that case what happens? Now I am showing this part, this is parental type, right? Now same F1 generation recombinant type I will show you here. Recombinant type. Now how will be that recombinant type? These are the genes. One Y plus W, another one Y and W plus. These two will remain same. These two which are there, those are same. Those are not getting exchanged. Y, W, and another one is something like this. Okay, these are the gametes of F1 generation. Now you cross it. You are crossing between these two. You will take this with this. What you will get? Y plus W and YW. Okay, then you will get same Y plus W and you will get a male. Now here YW plus YW, YW plus and a male, something like this. Just what is the difference is here there is no crossing over, here there is a crossing over. After crossing over you will get like this. So same one you will find here. These are the recombinant type. Here they have not shown that recombination which has been occurred. Directly they have shown here as a recombinant type. You can find Y plus W with a male, YW plus with a male. You can find they have given here Y plus W with a male, YW plus with a male. Right. Then what it has been shown? Y plus W with a YW, YW plus and YW. Now actually what is the point behind this? Right, what they are trying to explain on this. These are the parental type, okay, these are the recombinant type. Now in parental type they have given 98.7% are parental type and 1.3% is a recombinant type. Okay, so this is cross A. In a cross B you can find there is some difference. YW, here the distance between the genes you can find, and in the cross B the distance between the two genes you can find. Which is having more distance? That is W and M are having more distance between the genes. Here they are having less distance between the genes, right? So similar cross it has been done with a white miniature and a wild type. When they have crossed this, you can find similarly you can cross the same by taking pen and a pencil and cross it as I have shown in my note. So you will get the same one, and but parental type 62.8%, recombinant type is 37.2%. Now what is the difference here? When the distance between the genes is more, okay, here it is more, here it is less. When distance between the genes is more, recombination is more. Recombination is how much? 37.2%. When distance between the genes is less, then the recombination is how much here? 1.3%. Okay, as linkage will be stronger, when the genes are nearer, the distance between the genes is less, linkage is stronger. When the distance between the genes is more, that linkage is weaker. So parental type is less here, recombinant type percentage is more. Here parental type is more, recombinant type is less. Why it is? Because of distance between the genes. As they are closer, as they are nearer to each other, the bond between them will be stronger. So to break that bond, it requires more energy. So chances are less to undergo recombination. But here, when the genes are very far away from each other, then there is a presence of bond, but weaker bond, easy to break those, and easily it can undergo recombination. Okay, so this is what linkage and recombination, which has been shown in your textbook with this diagram. Now, to show that, here it has been given. Once again, I shall read out. Morgan and his group also found that even when genes were grouped on the same chromosome, when genes are present on the same chromosome, some genes were tightly linked. Is it? Some genes were tightly linked, which showed very low recombination, while others were loosely linked, showed higher recombination. Is it? Which were loosely linked, which are far away, which are tightly linked, which are nearer to each other. Okay, for example, he found that the genes white and yellow were tightly linked and showed 1.3% recombination, while white and miniature showing miniature wing showed 37.2% recombination. Okay, now this is the easiest way to understand this concept. And about linkage maps, how to study about the distance between the genes, we have vast information on that, but for your syllabus it has been restricted only for this, and this itself is difficult for you all, I think so. But for exam point of view, it has been asked what is linkage, what is recombination, it has been asked. Till now, the good news is that we have not got any question related to draw this. Okay, draw this, explain this, we have not got till now, but if you get any question, you must be able to answer about it. Okay, and not only this, even if they have asked about a student Alfred, a student of Thomas and Morgan called as Alfred Student, what does he get, who gave linkage map, okay, what is linkage map, about what is linkage, what is recombination, who gave linkage and recombination, this kind of questions, and especially entrance exam questions have been asked. 1 or 2 mark questions, that's it, not complete 5 marks on this. Okay, hope guys you have understood the concept. If you have any doubts, any queries, please comment it in the comment box. I shall meet you all in my next video. Till then, stay safe, stay healthy.